In this video, we are going to learn skeletal muscle tissue. Okay, this is one of the largest chapter in ENP1. So you have to, like any other chapter, you have to read the book first, and you should go over the PowerPoint slide, watch this video, and make sure if you do have any question, write down those questions so that we can discuss later on, and then that is how you prepare. And okay, so let me share the screen with you, the PowerPoint. Give me a second. So muscle are three types: cardiac muscle, skeletal muscle, and smooth muscle. These are the three types of muscle. Cardiac and smooth, both of them we are going to learn mostly in NP2. Skeletal muscle is the one that we're going to focus in this particular class. Okay, so after this slide, like I'm going to describe this first, and after this slide, when I say muscle, I actually mean skeletal muscle. Okay, so cardiac muscle and skeletal muscle, both are striated muscles. They have striations, meaning they have banded pattern, like exactly like my finger, like bent. Okay, or a checkered shirt. I'm going to give you, show you an example later on. So they have a banded pattern, like a light band, dark band, light band, dark band. We call them striations. But on the other hand, skeletal muscle is the only one that is voluntary muscle. That is the voluntary muscle, meaning you have conscious control of that muscle, like when you want them to work. Conscious control, like if I want my skeletal muscle to move my arm, I can do it. But on the other hand, cardiac and smooth, those two are involuntary muscle, meaning even if you do want right now to increase your heart rate, you can't. Your body can. That's a different story. Your body, like whenever required, they can increase and decrease the heart rate, but you consciously cannot increase or decrease your heart rate. So right now, if I want, hey, let's increase my heart rate, I will not be. If I do exercise, then when oxygen demand to the body raises, then body decides to increase the heart rate. That's a different story. So you have to understand this. So that's called involuntary. Or inner organ, like all the hollow organs, that's in your body, such as, to give you an example, stomach is made with, there are other things, but one of the important things is this smooth muscle. Now, if you do want, you can't really move your stomach a little bit because it is involuntary. So cardiac and smooth, both are involuntary muscle. Skeletal is a voluntary muscle. But skeletal and cardiac, both are striated muscles. Smooth is not. They're non-striated muscles. Okay. And I'm just going to mention one single word, which you're going to need it later on in NP2 a lot, because in cardiac muscle and skeletal, both are striations. But in cardiac muscle cell, there is something called intercalated disc. Intercalated disc. That intercalated disc is found only in cardiac muscle, not in skeletal muscle. Okay, but let's go to a little bit more in skeletal muscle. Skeletal muscle, again, one more time, voluntary, striated muscles, and obviously from the name you know, they're attached to one or more bones. Again, pay attention to this one. From now, when I say muscle, I actually mean skeletal muscle, because cardiac and smooth, they're not going to study right now. Okay? This is the pattern that I was talking about. That dark pink color, light pink color, dark pink color, light pink color. That is known as striations or stripe, okay, or striations or stripe or banded pattern. Okay. Now it is a voluntary muscle. Okay, now muscle cell, the skeletal muscle cell, like any other cell in your body, muscle cell, they could be very long. Not only that, they are cylindrical in nature. They're like a cylinder, you know, they are like my arm, right? Like this, cylindrical in nature. Okay, they're like finger, like this, cylindrical in nature. That's what the word, cylindrical. That's why it's a little bit long, it could be a little bit long, and it could be as long as 30 centimeters. For a cell, 30 centimeters is actually huge. That is why, now pay attention to this particular word. Muscle cell, what kind of muscle? Skeletal muscle. Skeletal muscle cell, sometimes known as skeletal muscle fiber. So when you see a word skeletal muscle fiber, it actually means skeletal muscle cell. So skeletal muscle cell, sometimes called skeletal muscle fiber. Okay. So before we go into their anatomy in detail, like microscopic level even, let's talk about their general function. So skeletal muscle involved in the skeletal movement, meaning bone movement. So when you move these things, we studied in our movement chapter that flexion, extension, abduction, adduction, all this movement is possible because the force generated by the contraction of your skeletal muscle. They can maintain posture, body posture and body position. They can support the soft tissues inside. They can guard entrances and exit. What does that mean? I'll give you one example. Let's say something, when you eat, it goes to the esophagus and then goes into, let's say, stomach. So right here, the junction of the esophagus and the stomach, you have a guard, like a rubber band. I'm using the word rubber band just to make you understand, like a skeletal muscle fibers, like cell, that whether something can go in or not. So they will open and close. Those are known as like sphincter, like acting as a valve. That's what they're talking about. Same thing, in urinary system, we have the exit. Skeletal muscle cell maintain body temperature. What does that mean? This is an indirect maintenance, meaning skeletal muscle, when they work, they require a lot of ATP to burn. And we know whenever you burn adenosine triphosphate or ATP, that's energy molecule, a lot of portion of it is released as heat. That heat helps maintain the body temperature. So that's like a general function. So now we are going to learn more anatomy. So we are going to learn first cytology, meaning like cellular anatomy, and then we are going to go into even microscopic molecular level in detail. Before we learn the individual cell, like how they look like, we must know that the bunch of cells, which is known as tissue, is organized with the help of connective tissue. So if we talk about a muscle, let's say I'm talking about bicep muscles. If I picked up this bicep muscle, literally I picked up this bicep muscle. So this muscle, this bicep muscle right now in my hand, that muscles is basically a bunch of cells, like a muscle tissue. So let's imagine we have 1,000 cells. So I mentioned earlier that each cell is like a cylinder, is a cylindrical in nature, like a little bit circular long, like this kind of cell in this picture. So look at that. This is, let's say, one muscle cell. If you imagine this is a muscle cell, so obviously we have thousands of them. So those thousands of them are organized with the help of 3 layers of connective tissue. Those layers are, first the name, innermost layer called endomysium, middle layer called perimysium, and the outermost layer called epimysium. Endomysium, perimysium, epimysium. Now learn this carefully because this is the exact same type of anatomy we are going to learn in nerve. Only the name will change. That time we will call it endoneurium, perineurium, and epineurium. Right now we focus here. So mysium means muscle. So endomysium, so it's an innermost layer. So basically, if you consider individual muscle cell, now right here, see, this is, let's say my finger is one cylindrical muscle cell. This individual muscle cell is surrounded by a delicate layer of connective tissue, and that is endomysium. So I have one muscle cell, 2, 3, 4, 5. All 5 muscle cells, they are individually wrapped by endomysium. Then we have another wrap around them, create a bundle. Then we have another wrap around them, create a bundle. This wrap, when they're creating a bundle, like 5, 10 cells together creating a bundle, that's called perimysia. And that bundle has a name, that bundle called muscle fascicle. So when it referred to the muscle fascicle, it is basically a bundle which is surrounded by perimysia. Now we have 1 bundle, 2 bundle, 3 bundle, let's say 10 sub-bundles, and then we surround them with another layer outermost, that's called epimysium. That's surrounding the entire muscle cells, okay? Epimysium and perimysium are mostly the area where lots of blood vessels and nerves comes, give the nutrition to the cell. So one more time, now let's look at the picture here. This is single cells. So all this white tissue appearing in this picture, meaning that is surrounding this, is known as endomysium. So endomysium, outside of the plasma membrane, they surround. So I think I have a better slide. Now let's look from here. So the tissue that surrounds outside the plasma membrane, that's called endomysium. And then this is let's say this is one cell. So we have tons of cells here. So when they create a bundle, like let's say 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 cells, this layer, like outside this layer is called perimysium. That's why this white layer is called perimysium. And then we have, in this picture, we have 1 bundle, 2 bundle, 3 bundle, 4, 5, 6, 7, 8, 9, 10, 11 bundle. And then we have something go around that, that's called epimysium. So epi is outermost, peri is in the middle, endo is in the innermost layer. Okay, so that's how our skeletal muscle cells are organized. So when you're looking at a muscle, you're looking at this bundle. Now we are going to focus on individual skeletal muscle cell. So individually, if you look at them, they appear like this. Individual, this is one cell, and they appear lots of red dots. These are nothing but like myofibril. That's a word. Myo means muscle, fiber. That's the word came, myofibril. So basically, if this is a cell, inside that cell, we have tons of longitudinal. Just imagine this. This is the example I want you to understand. So you have a circular, like a drum, a garbage can, circular. And if you put bamboo sticks, like hundreds of bamboo sticks in that garbage can, that's your skeletal muscle cell. That's your skeletal muscle cell, because garbage can, circular, appears like a cylinder, and the cylinder is filled with all these bamboo sticks. Individual bamboo sticks are actually, think about a muscle, a myofibril. So individual myofibril are those bamboo sticks. That's your muscle. So this, when you look from the top, this is how they are going to look like. So let's look from the side, how they look like. This is how they look like. So this is the muscle cell, like a cylindrical structure. And these are all these bamboo sticks. So they're showing a little bit bigger. I'm talking about individually this. This is like a bamboo stick. So in this picture, we have 1, 2, 3, 4, 5, 6, 7, 8, 9 bamboo sticks, and even more right here. So that is how a muscle cell look like, okay? So now let's go back to learn a little bit different way that what is skeletal muscle cell, what they call, and what are the additional feature, and then we are going to go to that detail. So first feature, skeletal muscle cell has multiple nuclei. Skeletal muscle cell has multiple nuclei. That's very interesting for a cell, because normally a cell has one nucleus, but skeletal muscle is a cell which has multiple nuclei. So it's extremely metabolically extremely active because they have multiple copy of the same genes. So remember that. Second thing, the stem cell that makes a skeletal muscle. So I'll give you an example to understand this. So let's say theoretically, one skeletal muscle cell, they have 50 nuclei, 50 nuclei. So basically what happened is 50 myoblast, those are the stem cell, they came together and they fused. Just imagine my example, 48 of them fuse together. So 50 myoblasts come, so 50 myoblast cells, 48 of them, I intentionally chose that number, so 48 of them fuse together. So now they create a giant cell which has 48 nuclei. That's a skeletal muscle cell. And then 2, remember total 50, so 2, that nothing happened, no fusion happened, those 2 unchanged myoblasts. So they are unspecialized myoblasts remaining with the big cell is called satellite cells. So those 2 unchanged myoblasts, 2 again is arbitrary number, we call satellite cells. So they are basically unchanged myoblasts. They remain with the same cell, we call them satellite cells. Their satellite cells' job is to multiply and produce new muscle fiber to some degree when repair is required. Skeletal muscle cells, another very interesting feature is you can understand the obvious reason because they are highly metabolically active cells, so they need a lot of energy, so they're packed with mitochondria, Now, skeletal muscle cell, their plasma membrane, like any other cell, we have a cell membrane or plasma membrane, they have a fancy name. We call them sarcolemma. The word sarco means flesh, lemma means membrane. So the sarcolemma, that's the other name of the plasma membrane of the muscle cell. Similarly, sarcoplasm, that's the muscle cell cytoplasm. So cytoplasm of the muscle cell called sarcoplasm. Sarcoplasmic reticulum. Remember endoplasmic reticulum? Same thing, sarcoplasmic reticulum. So they are basically modified ER. So what they do, their main job is to store calcium ions. Now remember, do not forget this. Calcium ion is required for muscle contraction, skeletal and cardiac. So we are talking about only skeletal right now. So calcium, without calcium ion, skeletal muscle cannot contract. So we need calcium ion. So calcium ion need a storage. So that's the reservoir. So sarcoplasmic reticulum, which is modified ER, is actually stores calcium ions. So this is a modified smooth ER. They store calcium ion with the help of a protein. That protein name is calciquestrine. So calciquestrine is a protein that helps storing of tons of calcium ion in the sarcoplasmic reticulum. In this picture, you see that blue color sac, those blue color bag or sac, those are sarcoplasmic reticulum. Inside this sarcoplasmic reticulum, we have calcium ions, which is required for muscle contraction. How they come out, how they go back, we are going to study that later on. So just to give an idea, like inside the sarcoplasmic reticulum, the calcium ion concentration is about like 40,000 times higher than that of the cytoplasm of the muscle cell. So in the cytoplasm, there is some calcium, but not that much. Sarcoplasmic reticulum has tons of them. So even for a microsecond time, if the sarcoplasmic reticulum opens to release calcium ion, that's a tons of calcium ions. We are going to need that later. T-tubules, or short form for transverse tubule, because they run like transverse, meaning almost like perpendicular to the plasma membrane. So this is the sarcolemma or the plasma membrane of the muscle cell. You see that yellow rubber band kind of look? Those are transverse tubule. So basically what they do is this. So let's look at this. Let's say this is the myofibril. Sarcoplasmic reticulum is like my shirt, like a bag. They go on top of this. Now, transverse tubule, they are tightly holding these things together. So they literally, if you look at carefully, so this is the myofibril, and that's the sarcoplasmic reticulum, like a bag sitting on top of the myofibril, and this rubber band-like structure, T-tubule, holding them tight. So when you're holding them tight, anything if you hold tight, the two sides of that rubber band will be swelled a little bit. That's exactly what happened. These two sides of that yellow line, you can see here, as I'm saying, rubber band, swelled or expanded a little bit together. That swelled area to both sides and the rubber band together, we call triad. So triad is basically whenever a T-tubule and circular myofibril, the tubule is tightly bound to that area. So that results is that external expandation, like, a little bit expanded chamber. Those chambers, these expanded chambers are called terminal cisternae. The word terminal means end. Cistern, the word came from the reservoir. So this combination of two terminal cisternae and the T-tubule is called tria. So that gives you a little bit the idea of what is inside, like some of the organelles and the membrane. Now the most important part. Those bamboo sticks in my example that I referred to are called myofibrils. Myofibrils is, if you notice, pay attention to this picture, you will see a blue color myofibril and a red color myofibril. Myofibril is basically made with two kinds of filament. One is called thin filament, one is called thick filament. Thick and thin filaments. Individually, they're made with some protein. So thick filament made with a protein called myosin. Thin filament made with a protein primarily called actin. So myosin is the protein which is responsible making thick filament. Actin is a protein that is responsible making thin filament, although detail of their interaction and their structure coming up later on. So this is a fundamental structure of the skeletal muscle cell. So now we are going to focus on a little bit extra detail of one particular myofibril and see how it works. That brings us to sarcomere. So what is sarcomere? This is the most important part of the chapter. Sarcomere is basically a repetitive small functional unit of the muscle fiber. Meaning, let's imagine this in your head. So let's say this is a myofibril. So my hand is extended form of myofibril. So when this one myofibril, so one bamboo stick, so when they contract, they literally make the muscle cell contract because those myofibril are attached to the plasma membrane on the side. So this myofibril is attached to this end, this myofibril attached to that end. So when they contract, they bring the cell like this, the cell kind of like a bunch. Okay, that's the concept you should have. Now, individually, this myofibril can have like 10,000 sarcomere. So it is like a, imagine this, it's like a wall made with bricks. So if you have 100 feet long wall and each brick, if you imagine like 1 foot, so you have a 1 foot brick, then 2 foot, the 2 feet, 3 feet, and so on. We lie down 100 bricks to make a 100 feet wall. Now imagine if each brick becomes slightly shorter, the whole wall will be shorter. That's your myofibril. That's exactly how it works. So each myofibril is nothing but those individual made with 10,000 those brick. So each brick is the sarcomere. Sarcomere, that's a unit. So that is why sarcomere is known as the functional unit of skeletal muscle. So now, in order to understand how a muscle cell contracts, you don't need to study the whole muscle myofibril. You just need to study one sarcomere. It is exactly like same way. If I say, okay, what is the characteristics of the wall, then you don't have to study the entire wall. You just study one brick. So if you can understand what is the characteristics of that little brick, you know what is the characteristics of the wall. Same thing here. If you know one sarcomere, how it works, you know how myofibril works. So let's focus on one sarcomere now. So one sarcomere, what they made of? They made with thick filaments, they made with thin filaments, they made with proteins that stabilize the structure, the position of the thick and thin. And number 4 is they have a protein that regulates the interaction of thick and thin. So those are the 4 major things of a sarcomere. So sarcomere has thick filament, thin filament, obviously those 2, and then there is a group of proteins that make sure their positioning is right, that is structural, and there is a group of protein that make sure their interaction is accurate. So that's together is a sarcomere. And sarcomere is functional unit of skeletal muscle, because if you understand how each sarcomere contract, then you will understand how whole muscle cell contract. This is a structure of a sarcomere. So the sarcomere at rest is the top one, and sarcomere when it is contracting, the muscle fiber contracting. So let's look at the sarcomere on the top one first. Okay, now pay attention. This is a schematic diagram by the artist. Okay, so now all the lines here, all of them, all the lines here are protein molecules. So we are now at a molecular level, or molecular structural level of sarcomere. Because without understanding this, we will not understand how muscle cell work. Okay, so at this level, again, remember everything is protein here. So now look at the first, this dark blue color protein. That's right here. Follow my pointer. So those dark blue line, those dark blue color protein are called thick filament because they appear thick. Their density is more. They appear thick under the microscope. That's why we call thick filament. And these red lines right here, that's horizontal red lines, those red lines here and those red lines here, these are called thin filament, because again, their density is less. Okay. Now these are the two main structure. Now these two filament, how they're organized with the help of other protein. So let's look at the first protein. This particular one, see that kind of like, I'll give you an easy example to understand, like a zigzag. This protein, it's called Z-line protein. Z-line protein. They are made with a protein called actinin. So right here, I wrote down in my slide, Z-line is made with a protein called actinin. Don't do mistake with actin. Actin is the thin filament. This is actinin, which is the Z-line protein. Then right here, so Z-line protein, let's go back to Z-line one more time. So Z-line protein holding those two sides, like this is a sarcomere, this is a neighboring sarcomere. So this is a sarcomere and that's another sarcomere. So Z-line protein, they are attached to the thick filament, thin filament right here. Those red lines are thin filament. So Z-line directly holding. So Z-line directly holding. So if this is a Z-line, this is a Z-line, this protein will be thin filament protein. They're holding directly like this. So right here is a Z-line, right there is a Z-line. Between two Z-line, that's one sarcomere. So a sarcomere is between those two Z-line. So here's a Z-line, here's a Z-line. That is a sarcomere. That is a sarcomere. So right here in the middle, those are your thick filament. But again, they can't just float. They have two support. One support right here is a huge protein called M-line protein, like a middle line protein. So M-line protein right there. And there's a protein right here. You see that green color spring kind of structure? Those are called titin, T-I-T-I-N, titin. That's a protein. That's an elastic protein that connects thick filament to the Z-line. So if you go back one more time, so this is M-line protein right in the middle. M-line protein, they're connecting all these things. So all these things vertically, this vertical protein connecting all these thick filament. And there is a protein right here. Those are called titin. They're holding the thick filament on the side. So this is the structure. This is a structure of a sarcomere. Now imagine this. Okay. So first of all, we have to understand the structure under the microscope, like how it looks like. So when you looking under the microscope, pay attention again. This area from here to here, this area, they appear dark because they are high density protein. So from in this area where it says A band, they appear dark. So from here to here, that's called A band or dark band. Because in this area, not only you have a thick filament, but you also have a part of the thin filament. So what is A band? A band is the entire length of thick filament and partial thin filament. Together, that area become very dark. That area from here to here, we call it A band. A band, okay. From here to the next neighboring sarcomere. So this is another sarcomere, remember, from this Z-line to that Z-line, another sarcomere. So from here to the neighboring sarcomere, that portion is only the light dense or only the thin filaments are there. So this area we call light band because comparatively a less dense area. So this is called light band. So the light band is basically, we can imagine the beginning of we can say from this side, the end of the thick filament to the beginning of the thick filament of the next sarcomere. So that's your light band. That's your light band. So we have two bands, one from here to here, A band, one from here to here, light band. So half of the light band belongs to this sarcomere, the other half of the light band belongs to that sarcomere. So those are the two light band. But this is how you can memorize. I wrote down here in my slide, like A band that contain partial thin and the entire thick filament, it's called dark band. See that second letter A, and same thing here, light band, the second letter I. So I band is a light band, and A band is the dark band. You can memorize that way. It's like a coincidence. Okay. Now, if you go back to the previous slide, you'll see this area right here in the middle. This area sometimes referred to as H band or H zone, because this area is a little bit different than the remaining part of the thick filament. The entire thick filament is not same dense. This area is slightly less dense because they don't have this additional thin. They have the thick, but they don't have that additional thing. So they have the blue line, but they don't have the red line here. So this area is slightly less dense compared to this portion. So this portion is called H zone. And this part where the thick and thin filament go side by side, like this part is called zone of overlap. This part is called zone of overlap. So H band is a lighter region where no thin filament are present, only thick. So that's the only thick area. And zone of overlap is where both thick and thin filament are there. So both ends. So if we go back one more time, this is the H zone, and that's the zone of overlap. In the zone of overlap, they have both thick and thin filament present. Now, one very important thing, if you look at the three-dimensional structure, the one I taught you from, it's two-dimensional. If you look from the side, three-dimensional structure, each thick filament is surrounded by 6 thin filament, and each thin filament is surrounded by 3 thick filaments. So those who have a chemistry background, I'm going to just mention here, sodium chloride lattice structure. If you do remember from your chemistry class, you will understand that. The rest of the class, I would say right now, you can memorize that sentence. Basically, each thick filament is surrounded by 6 thin, and each thin is surrounded by 1, 2, 3 thick. So that's how they organize the entire thick and thin filament. Okay. Now let's go back one more time. Now, if you imagine the top picture, if you put a hand here, and if you put a hand here like this, and if you press those Z line, if you press just imagine this, if you press those Z line on the two sides, the band is going to go move like this. So the partial thin filament is going to go like this because there is an area here. So the picture will look like that. So if you put a force on this side and a force on that side, the whole thing will shorten, contraction. The whole thing will shorten, that's contraction. So this is actually called sliding filament theory. So as if like the filaments are sliding past each other. So this is the zone of overlap, boom, they move inside. So that's called sliding filament theory. Now what is happening in the sliding filament theory? From the picture, you can easily figure it out. First of all, A band, which is the entire length of the thick filament, remain constant. So that's point number 1. A band remains constant because there is no change in the A band. But look at the I band. I band was huge. Now I band decreased, becomes less. So from that much area, it becomes this. So the second point of sliding filament theory is I band shorten, or I band reduces their size. So A band remain constant, I band shorten. Now look at the H zone. H zone was huge here. Now look at the H zone here, they're less, so we can say H zone reduces. The H zone was this much, H zone becomes now this. So H zone reduces. And look at the zone of overlap. They were here, now they're even longer than that. So this much become that much. So we can say zone of overlap increases. These 4 things together is the summary of sliding filament theory. So A band from here to here remain constant. I band from here to here get shorter. H zone from here to here, they get shorter. and then finally zone of overlap from here to here, they get longer. So those four together is the actual sliding filament theory. So let me see if I wrote down that. Not here, but you can find in any book when you study those 4 points they're going to mention. This is the electron micrograph, by the way, they use the electron microscope to look at the sarcomere as a whole. So let's look at them one more time. So these are those Z line or Z disc. We call them Z disc if you look at the three-dimensional structure. So between those two Z line, that's a sarcomere. So this portion is the I band. See that this is a neighboring thick filament. That's a neighboring thick filament. If there is a Z line right here, there'll be another sarcomere. If there is a Z line right here, there'll be another sarcomere. So this is I band, so which gets shorter during sliding filament theory. That middle area where there's a slightly light color, that's H band. And at the middle of the H band, we have the M line protein that are holding them together. And the A band, or even the sarcomere becomes shorter, A band remain constant. That's sliding filament theory. So up to here, you know exactly like how a muscle cell organized, how a skeletal muscle cell looks like, their microanatomy, and how the skeletal muscle, like how the myofibril look like, even their molecular level of anatomy, okay? And the second part, we are going to learn like how muscle cell, then how the sarcomere that we just said that they do undergo like this, like we say the sliding filament theory, how it actually happened. So that's the next part of the lecture. So let me stop right there, and then I'm going to teach you the second part on another segment. Okay.